Good afternoon. The topic of my today's lecture is the feeding of laboratory animals with a very special concern of the microbiome of the animals. As you know, according to the black box hypothesis of the experimental design, the animal can be considered as a black box receiving different stimuli. In order to assure that only our stimulus should be different in the experimental and in the control group, we have to standardize all the other environmental circumstances like the genotype, the keeping, the environmental humidity and temperature, and of course the feeding of the experimental animals. This lecture was prepared for the 4th East Mediterranean Congress of Laboratory Animal Science, organized online from Istanbul University by the Turkish Laboratory Animal Science Association. Concerning the feeding of the laboratory animals, the first problem we meet is that we have great differences in the species and strains and digestive systems. You see that some extreme examples can be given, that the Nude mouse, the Spiny mouse, or even the non-human primates, the Marmoset monkeys, may have a absolutely different type of digestion and of course feeding. But at the first glance we can make a classification according to the main feed sources, like herbivore, like rabbit, guinea pig, omnivore, like rat, mouse, non-human primate, pig and poultry, and carnivores like dog, cat and ferret. The original ancient digestive type and digestive system used to be similar to the mole digestive system, namely it is a insectivore digestive system with a big stomach with two parts, and a very simple small and large intestine, and with only an apparent cecum. If you see the development of the different digestive systems of the possible experimental laboratory animals, you can see the dog digestive system with a relatively huge stomach, simply intestine and very small cecum. This is a slight carnivore, approaching to the omnivore. And here you have a omnivore digestive system of the rat. You see that in real life, left side and in picture, what is interesting compared to the carnivores, that we have a huge cecum which will allow the rat some efficient fiber utilization, fiber fermentation and fiber utilization. Another omnivore type related to the pigs and mini pigs with a medium-sized stomach and very, very long small and large intestine, medium-sized cecum, and what is not on the picture, but I can add that a very, very active pancreatic enzyme activity included the starch-splitting amylase. And here you have the digestive system of the ruminants with the rumen and the other part of the forestomach and very, very long small intestine and medium-sized cecum and colon. Now we will concentrate on the feeding of the glires, namely the rodents and the rabbit. But before that we have to see the phenomenon of trophallaxis, which is common in the glires. Basically, the first signs of the trophallaxis can be found in ants. The definition of the trophallaxis is the use of own or species companion biological product like feces, secrete, saliva, urine, etc. for feeding itself or each other. The examples is the ant kiss, when they change, they vomit some content of the stomach to the other, but also the coprophagy of the rat, the zygotrophy of the rabbit, the crop milk production of the pigeon, and the vomit puke of the canidae, especially the wild dogs. Here you can see some illustration of that. The rabbit has two feces types, and the small and yellow is high in fiber, is excreted separately, and the cluster-like soft feces is recycled directly from the anus. Here you can see that the guinea pig and the chinchilla has also two types of feces, and the black one is recycled in case of fiber protein deficiency. Now the question arises that how to determine the nutritional needs of the laboratory animals. The criteria of the requirement should be similar to the production animals, the growth rate and the production, milk, meat, wool, etc. No, absolutely not. In case of laboratory animals, the criteria of the requirement for energy concentration, protein concentration, etc. is the reproductive performance and the longevity of the animals. The later will allow the human extrapolation too. The formulation and labeling of the feed may be an open formula and closed formula. It means that the closed formula is generally accepted, but this is not the best solution, because if it's closed, the natural ingredient from our eyes, we cannot consider, for example, that soybean is used, for example, with genistein estrogen, plant estrogen. So we would prefer the open formula, giving not only the energy and protein and calcium phosphate concentration, but also the natural ingredients. Yes. And using this evaluation of the nutritional needs, here you can see the nutrient requirements of the rodents. The energy is given either in digestible or metabolizable energy, and the range is 13 to 18 megajoule per kilogram feed. The range is large, but it's no problem because from one side the voluntary feed intake, and from the other side the coprophagy, psychotrophy may help. So these animals basically are eating according to the energy concentration of the feed, but the guinea pig gives exception, and this is the reason that the guinea pig using ad libitum feeding may get obese. The protein range of the lab diets is between 18 to 22% crude protein, which is equivalent to 12-14% of ideal protein. In some cases it's given. The range is also large, but by means of the coprophagy and the psychotrophy, the animal can adjust the amino acid supply to the diet. As a specialty, the fiber needs are different. Some fiber needs is obligatory because of the dietetic effects of the dietary fiber, but it is extremely different. High fiber needs can be found in rabbit, chinchilla, and dagu, and of course the horse, and low fiber preference can be found in case of the hamster. Here I give an example how to indicate the nutrient concentration in a scientific paper. You see that it's given the main characteristics like 5% weight per weight fat, 13.5 megajoule per kilogram energy, or you can give a reference diet here, which can be found in the internet, etc., etc. It is extremely important not only in the scientific papers, but also when you are submitted a paper for ethical allowances. The nutrient requirements can be modified by the strain. Of course, that the hairless, obese, or nude mouse has a different basal metabolism, different energy requirement. Of course, that the microbiome, which is important today, has a great influence, especially to the vitamin supply and to the others. See later. And very important that many times we need a heat treatment of the laboratory diets, which will degrade in a different degree according to the different vitamins and the minerals, mostly the vitamins. A specific feature of the guinea pig and the fish that they cannot synthesize ascorbic acid, and lack of ascorbic acid, they will show vitamin C deficiency syndromes like shorter bones and scurvy, hemorrhagic, and scoliosis, lordosis of the spinal cord. The lab diets may be classified into three groups, like the natural ingredient diets, the purified diets, and the chemically defined diets. During preparation, manufacturing of the lab diets in the practice, we need separated facilities. We cannot use antioxidants. We prefer a very low or no microbes or mycotoxins. from the minerals and vitamins, we have to choose the good availability group. To assure the homogeneity, we have to watch the mixing time, avoid auto-segregation, and we have to give a good storage. The physical form appearance of the diets is very variable and much more than in case of the production animals. We have the meal or coarse flour, maybe have a cake too to avoid the auto-segregation. Pelleted with different measure and with different binding material. Crumbled for small animals, extruded, see later, baked, flaked, semi-moist or gel form, and even liquid when we can use filter sterilization. Here you can see that how an extruder is working by forcing wet meal mixture through a die under high pressure and temperature, which results in expanded product at the end. Especially pet, fish, and ape are fed on extruded diet. The feeding and the drinking technique generally is ad libitum, but in this case the spontaneous tumor development and the decreased expected lifespan can be found. So it would be better to make a rationing. It is important the water supply, and we have to consider the circadian rhythm. Why? Because during ad libitum feeding we have a low amplitude of different circadian and metabolic rhythms. In case of the time restricted, a high amplitude, which is beneficial against adiposity, glucose intolerance, leptin resistance, liver pathology, inflammation, and motor coordination, and improve the motor coordination. But this cannot be used only in smaller experimental groups and not in the breeding, of course. Yes. Energetic rationing is important. It means that the protein, amino acid, minerals, vitamins, and especially the test compound can be related, should relate not to the dry matter, but on the energy unit, which will avoid the different intake from the test and from the test compound. Feeding and welfare are also related. Welfare and environmental enrichment is important because work for feed can prevent the stereotypies if the animal need to search the feed. And what about the treats? Okay, treats can be given if in the frame of the experiment can be allowed. Especially you can give natural feed particles, slice of mandarin, whole ground nut, seed, or novel food items can be found. You can see that compressed into holes in a wooden block or in compressed in a pasta shell or freeze-dried feed sources can be given as a treat. how to feed during transport and acclimatization. It is important that you have to know the species characteristics. For example, the pig and mini pig should not feed during the transport because they vomit very easily. Inversely, rat and mice need feed and water. For these purposes, one can use a solid drink, which is also used in barrier in case of recovery after surgery and, of course, during the transport. Common metabolic troubles and diseases should be mentioned, like the rancid feed will cause oxidation and damaging of the some sensitive tissues. We may have mineral vitamin deficiencies. We may have iatrogenic dysbacteriosis, peroral, especially guinea pig, urolithiasis. Susceptibility to mycotoxin is extremely high owing to the coprophagy and psychotrophy, and the formation of hairballs or trichobezoar is also possible. Now, as a last section, feeding brain stress microbiome relationship should be overlooked. You see that the gastrointestinal tract may have 350 times more genes than the mammalian, in this case human, genome. And if you see that according to the microbiological standardization of the lab animals, we may have germ-free, mono, oligo, or poly-associated. We may have specified pathogen-free and conventional animals. So very, very different in microbiome. As a plus, the stress may have a huge influence on the gut-brain axis, namely decreases the permeability of the gut wall against antigens and pathogens. The stomach emptying may slow down. There is an inhibition of the hippocampus proliferation, and the oral tolerance get insufficient, which gives a danger of food allergy. And of course, generally, the digestive processes are disturbed. And now we see that the brain-gut axis, it is a there and back route way by mostly using the nervus vagus. The number and diversity of the microbiome is increasing. It is not the same in the gastrointestinal tract. Caudal, it will increase. You see the different genera of bacteria. And here you have a summarizing figure of the main functions of the microbiome. You see that it is not only part of the digestion. This is part of the modulation of the bone mass density, promotion of fat storage, angiogenesis, development and training of the immune system, basically important, biosynthesis of vitamins and amino acids, metabolism of drugs, modification of the nervous system, breaking down food components, resistance to pathogens can be formed, and there is a protection against epithelial injury. So these are shortly the functions of the microbiome. The microbiome may have an influence on the length of the villi too, by means of the produced short-chain fatty acids like butyric acids. But if there is a disturbance in the microbiome, we can have pathological consequences like arthritis, the composition of the peripheral blood concerning the immune cells will change, allergic inflammation in the lungs, type 1 diabetes and pancreatitis in the pancreas, and multiple sclerosis in the central nervous system, only to mention the most important. And here I give some very interesting publication showing the role of the microbiome. You see that the microbiome is different in the obese and in the lean persons and mice. And the germ-free mice are resistant to the high-fat diet, no obesity develop. And at the same time, the moderate stress may prevent enteritis through the change of the microbiome. Or the fecal microbiome can be transplanted and will, from an irritable bowel syndrome patient, and by this way we may alter the gut function and even the behavior of the recipient mice. Or undernutrition in childhood may be influenced by the microbiome, and enterotoxin producing Bacillus fragilis concentration may be at the background of this problem. Or the intestinal commensal bacteria may improve the mucosal immunity in the lung. Or a mineral vitamin deficiency will disturb the microbiome. And the human microbiome project is very developed, and some products are before marketing against diarrhea, eczema, ulcer, vaginosis, and ammonia trapping. Even the equine metabolic syndrome is caused by an abundance of phylum Verrucomicrobia, and the change of the concentration of Phyrobacter, Lactobacilli, and Ruminococci. Very similar can be found in human and mini pig too. It is difficult to establish. It is difficult to establish which is cause or consequence. This is the this is the hen egg dilemma, and the homogeneity diversity extrapolation, which is the most important in an animal trial. Hygienic standardization in the old time, Lathrop, very important person in the laboratory animal science, told that I can do my experiment today because my rats all dead in some diseases. And today, hygienic standardization according to the FELASA 2010, and I can do my experiment because my mice are too clean and there is a limited diversity of the microbiome. You see left a cecum of a germ-free animal and right a SPF animal. Great differences. So Lawkins and co-worker propose to standardize the microbiome, making a germ-free animal, and after giving 15-16 standardized bacteria to revitalize the microbiome. In my view, in a very fine experiment, we need a mosaic population concerning the microbiome. This is much more use of experimental animal, but in a very fine trial it can be founded. Summarizing my speech, the most important statements that the nutrient requirements are evaluated using the criteria of longevity and reproduction. The diets may be natural, semi-synthetic and synthetic. We have 8 physical forms. We have to consider the breed, strain, lack and presence or lack of microbiota. The feeding is practically ad libitum, but would be better rationing. The consideration of the circadian rhythm is important, and we have to consider also the influence of stress and microbiome. Thank you very much for your precious attention.